Oh, we can. A little bit close, actually. You're coming. Oh, I thought it was right. I wasn't sure if I had to make sure that what was over first. I sent her a message. So we like, can I come on? Or is it just only not your number? No, come. So, well, there's Facebook newsletter, so the 10 people are coming. So I guess she's doing good. She's leaving for Florida, but she's still in school. So her boyfriend and family are taking her. Well, that's taking her to Florida now. She's driving. Don't know why that. Well, she's in Indiana right now. That's the baby.
Hello, Marlene.
Hello, hi, How are you? Sorry, I heard that's going.
Yes. That's about the front spot.
Setting up the live stream.
Okay, thanks.
Because we're kind of like right outside.
This is the famous classroom.
Hi, well, my guys have a lot of So, no, it's not.
You gonna sit out there?
Maybe that. I've only watched these. You want me to go?
No, it's runs my eye. I can't see nothing.
Do you want me to go with all of you?
Sure. I just want you to take
I think that is something that's.
Yeah. She's just so.
I know, I missed it.
I'll make sure it's.
All right,
I'm sorry.
ready? 1, 2, 3. 1, 2. Okay, one more. Yeah, it's part of it. Yeah. It's probably not that. You can see it's not.
Sit there.
Hang on there.
Yeah.
There, we sit here.
What?
With Jason. Driving all over.
I did it already.
I
I did. I'm done with that until next week.
Why next? Oh,
Christmas.
you're gonna pick up people for that too?
No, I still visit them. They don't come. It's not at 1,200 miles, though, so that's.
Yeah. Then you take this. You, when you, driving?
Yeah, so we'll go to Memphis for a week.
You go visit friends.
So I think at least once.
You gonna need some bread?
Probably. What's today, Friday?
It's on the spreadsheet.
The drive's just sandwich.
2 days. Like, first of all,
As we can celebrate.
Right.
My mom's coming out here. Christmas Eve. There's my dad. She's been here for the last 2 years. Wait, let me go to the picture. But I don't want to otherwise over that part. Cynthia usually has something else. That's what's going on. Watch that.
So maybe it takes you like yours.
We got it. We got it.
Oh, we have 30 minutes down. This is a light.
We have it there.
Have you?
For the first year, because I used to go there for English.
Oh, yeah.
We'll have to take more than 2 people. Yeah.
So again, there's tiers. So like last year.
Yeah.
No way I can do it. So you can do 3 times a day.
Baseball, yeah.
You know, we can do as much as you want.
That's the way
Yeah, but even if your friends, unless you're best friends. So then there's
Give it to me. It's the laptop charge or something. This is just emergency. 83%. I didn't think you could find We'll try it.
Mine didn't start yet.
You guys rolled hats with this. Imagine that.
What that?
Rolled hat isn't something you've done before.
I feel like
Look at that.
there's always something to set it up for you. I just don't like
Such a good idea. I can't see. Whose computer is it?
Who's your internet?
What?
Are you asking me?
Yeah. That'd be
Are you looking for Google Chrome?
So were you at surprise or?
Yeah,
Yeah.
she knew already. Where'd Abby go? It's okay.
Yeah.
Close enough. Yeah, you heard the show me freaking.
Well, she blew it and she sat down.
Where did Abby go?
So, yeah,
You were on the receiving end
Yeah.
for sure.
Yeah.
Where did Abby go? Oh, where'd you go? Take my pointer. Oh.
Shook hands with your dad.
Hey, how's it going? Good. Where should we go? Right here? Who's in this one?
No idea.
Yeah.
I got it. No, they came from LA. Yeah. I got it. And when you maybe you might get here. So we're counting it as a whole.
I need to check my phone. Wait, why are you putting it there? For live stream? Are you maybe? Kind of. Depends on how I use it. I will hide behind the podium. Yeah. Okay, I will let you.
The ghost of Christmas past. Did you know you were going to be in it? Was that surprising for you? This is the But everyone down here is
Hey, nice to see you again.
See you again. Her attendance is awesome. She made this. Don't look. Stop looking.
What?
30 or 40. Just 4 minutes of really good not dressing. You can write a Yes. Wait. You want this?
You did it? You were like, what?
30.
That's priority yourself.
How are you, Jason?
You?
Happy?
Yeah?
She's
Excited?
almost there, right?
Yeah? He's here right now.
Oh, she's here. I'm talking about there.
Yes. Oh, San Francisco. Dude.
She's just 100% moment.
99.
Yeah, she needs to be level there for herself.
Yeah. Maybe. And then love you. Did you need anything?
I don't think I could keep it in.
My mom is super nervous for it.
Why?
Like my stomach's in knots, and so I was like, she's gonna be fine. She's like, where's the screaming link? You can be on YouTube, but.
I know I see it.
So he needed to set it up. You can be internet famous. Glad you passed me with this important responsibility. Thank you. Turn around. Star Wars?
But you can close out of other programs.
There's a
Does it work if you change it to mirror?
I can look inside and walk out. This is a what?
Who's here? Who are you? Hello? No.
So they should be able to hear, although it's very loud. There's 3 people watching.
Who are they?
They can comment if you want to see it. There's a chat.
Who are you?
So Bailey should be watching. He requested it specifically.
Hey.
Oh, Kwan's also watching. There we go.
Darren.
What? You can see who's on. Just try to log in.
No, it won't be fun. I just finished my acknowledgements moments ago, and I need to look at my files. No one will let me look at them. It's very stressful. No one gave me a shot before. Why is that? Why they get quiet?
Okay, we are having a little bit of technical issue. So we are trying to. Sorry. Oh, no.
Special guest technician. We're talking about if you don't work it out, you're just going to What is board this morning? I don't want to get caught off guard.
Is that your grandma?
Yeah.
She's so cute.
I know she is.
Back up.
I acknowledge you. Oh, you mean in the presentation? I don't know. Maybe. I might have a car rating fund. They're having technical difficulties. I'm talking to my invisible audience.
Quant.
Yeah. He wants it?
We're gonna do it on Instagram and switch it. No, if that's okay. Just load all your stuff. We tested it there. I'm gonna go through it.
It's happening. My phone. No, my computer wants to send it.
Oh, really?
Why do you
It's not gonna go.
the thing is, this is sad. We don't know what's
Okay, I think that we have the technical issue figured out. Okay, so thank you for your patience. So welcome to Reyka's thesis defense. And instead of telling you how much I'm going to miss her, believe me, I will. I'm going to show you a video from the lab. The video will start with a welcome email I sent to Reyka on March 31st, 2014. Let's watch.
As a leader, Reyka has magic power to solve all the conflicts, make things better, and share the world. And as a friend, Reyka can always provide you positive energy and make you to take immediate actions, even though friendship is strong. A part of her hair, like here, together because it makes things better. And you can never say no to Reyka.
Congratulations, Reyka. Wish I could be there. I'll be watching from my desk today. I once heard that you can tell a lot about a person by the way they treat those who can do nothing for them. And by that standard, I think you are incredible. Thank you for making the team what it is and what it will be. You really made it feel like a family. And again, I wish you the best of luck today. And again, congratulations.
Congrats. I'm sure others will comment on how smart, hardworking, friendly, and fun you are. I would add that feel, excel, and boast by mathematics and experimentation like you do. I'm also very impressed by your organization skills, conference calls, and many other occasions. You are quite aesthetic as well. I still remember vividly how you did one-on-one and score in trivia house. Sorry, Mike. That's also as you die. You're always very responsive. We'll miss you a lot, even if just for that. And we truly wish you the very best.
Reyka was one of the first people that I met at the interview weekend. She might not remember. And we know she turned out to be such a great friend. She is very helpful in the lab discussing science, but also very fun to talk to about other life and party events. We will definitely miss her in the lab, and we're really happy to see her graduating. So congratulations, Reyka, we'll miss you.
Yeah, congrats on getting your PhD. You well deserve it. I'm so grateful of having you back at my WashU days. You were such a strong ties to all our friends since the beginning of our grad school, and then we're the day lab, and it's just been so much fun with you. So I think Cree can help me so much. All right, we're now Cree.
So the thing about Reyka is I'm really glad that we ended up being in the same lab. I'm not used to having or working closely with a best friend on anything academic, but it's been great because I got to bounce off ideas and gossip with her about whatever was going on in the lab. In addition to that, she's just a really well-rounded person who cares about other people and does things very quietly without looking for any kind of recognition. She's very passionate about her science and her volunteer work and helping other children through YSP learn about science and love science as much as she does. And also, she's just a lot of fun, and she can balance her work and go out drinking, and I feel like she hasn't changed that attitude for any part of her grad school, no matter how stressed she was. And I think she doesn't sleep, but she's super human, and she can balance everything. And I can't even imagine what St. Louis is going to be like without her, and I'm part of the teams.
One day I come to the area, I was surprised to say Reyka was there also. Then I come to say hello to Reyka and ask her one question. To Reyka, did you stay all night in the lab? And she said yes. So Reyka is just such a dedicated person. That's what you can imagine from the conversation I had with her recently. She can make great things happen, both in life and research.
Hey, Reyka, congrats on making it to the presentation.
With your time in our lab coming to an end, I thought I'd reflect a bit on the impact that you've had in the lives of others. You've come to remind me in so many different ways, and you're going to be so nice. As far as I'm concerned, you know where everything is, you know everything that's happening, and I don't know how I'm going to know anything going forward. I've really come to admire your intellectual curiosity about so many different things. Not just science, definitely science, but beyond that, your curiosity about people and their lives and their relationships. Sometimes even their door codes. But beyond just being hardworking and smart, you're also kind and generous with your time. You really make an impact in the lives of others and in the community around you. I myself will forever be grateful for the time that you devoted to the Balsa Foundation. Reyka, thanks for being awesome. It was so good to have cross paths with you. May you do great in the future. May you soar far and high.
The video and the audio really speak from our heart. And Reyka, you had a terrific 5 years here. We will miss you. And you are not just a student, I'm very proud of. You're also a colleague and a good friend. With this, I'm going to turn the podium to Reyka.
Okay, I guess I can follow that maybe. So I'm excited to tell you guys a bit about my work, looking at these splice site creating mutations across cancer genomes. So our lab studies how cells change from a normal cell to a tumor cell. And it's commonly understood that cancer is a disease of the genome, whereby you get genetic changes that change a normal cell to a tumor cell that change it to alter how it grows. So it grows uncontrollably. And in order to understand that process, we first need to go through the central dogma, whereby DNA is transcribed into RNA and RNA is translated into a protein. And to go into that in a little bit more context, here we have the DNA sequence written on top. The DNA is broken up into exons and introns. Exons are these regions highlighted in red that are the coding regions of the genome that are put together through alternative splicing. Introns are removed through the splicing process, and these red regions that are exons create the RNA that is then translated to a protein, which are indicated by these circled letters here. And usually we try to classify mutations based on the change on the protein level. And here are just a few examples of what those changes could be, but I'm going to go into a little bit more detail on that in a few slides. So going back into the context of how does the cell change from a normal cell to a tumor cell, we like to think of what changes in the genetic environment. So generally at the start you have your genetic material of your normal cell has germline variants and other types of changes in the genome that are different person to person. But you may have just normal variation that doesn't really do anything, but just different person to person. Or you could have germline mutations that predispose you to getting cancer. As the cells evolve and different biological structures are involved, you may get changes that lead to initiating events in the tumor that cause changes that lead to tumor growth and development. As, again, biological structures are involved, you may get secondary and tertiary events, and all of these different types of alterations lead to the tumor cell development. So over time, as these cells are changing with treatment or other outside effectors, you can use different biological measurements such as DNA, RNA, and protein to try and evaluate what are these changes that are happening to the genetic material. For example, if we were to look at our initiating mutation, we could see that at time point A, it may be very low because it's not really present at a high frequency in the cancer cell. But as it moves on over time, you may see that it becomes higher. And even after treatment, it may decrease. So in our lab, we try and understand what are these different biological measurements we can use to understand how the tumor cell is developing. So going back to what these types of mutations are that could be these initiating events, here we have the DNA sequence that is indicated in the wild type form, and below here is the mutant form. So we try to classify mutations based on the changes on the protein structure, because the protein is what does all the work in the body. So likely these mutations that are leading to tumor formation are disrupting how that protein functions. So this first example here is the C to T mutation, which leads to a stop codon. Now stop codons are normally at the end of a protein, but if introduced earlier in the protein structure, it could lead to altering how that final gene is translated. There's also silent mutations such as the G to T, and that does not change the resulting amino acid sequence. And it's assumed that these mutations do not confer a change on the protein structure. There's also deletions where you get a removal of a part of the genetic sequence. That could change the frame of the entire protein. You could get missense mutations such as the C to A, which changes just the amino acid sequence. And then there are also splice site mutations, which are mutations at the GT and AG residues that are just adjacent to exon-intron boundaries.
So these sequences are highly conserved and are important for bringing together the coding sequences, or the exons, and removing these intronic regions. So a particular mutation type that I'm interested in is mutations that disrupt these GT and AG residues. These are highly conserved across many genes, and if mutated, can completely disrupt how we put these exons together. But what I want to know is, are there other types of mutations within the coding sequence that can act like these splice site mutations? For example, this silent mutation here has a GT because of this change. And is this GT accidentally utilized by the cell for activating this new hiding splice site? And what that would look like is, for example, here, this is the wild type sequence. We would expect this exon to be utilizing this splice site, but instead, because of this mutation, the mRNA may only read up until this region. And the splicing machinery may say, oh no, I'm going to use this site instead when mutated, and this will completely shift the frame of the entire protein and give you a different file. And this is really an important process to understand because splicing is fundamental to gene transcription and translation. So how this is disrupted in cancer is important to know genome-wide. So splicing normally happens within different tissue types, but you can get alternative transcripts that have different isoforms. And deregulation of various proteins can lead to all of these different cellular processes that are deregulated because of these alterations. And these are always dysregulated in cancer. So I really want to understand, are there mutations that are not properly being annotated because they disrupt the splicing process? And so for my talk, I've broken it up into two parts. For the first part, we're going to focus on somatic mutations. These are mutations that are present just in the tumor cell and not in the normal tissue, and understanding which of these are altering the splicing process. And then the second part of my talk, I'm going to focus on the germline mutations. These are mutations that are very rare in the population and are present just in a small percentage of people and are maybe predisposing people to cancer. And I'm mostly focusing on mutations that are overlapping the GT and AG site. This is the donor splice site and the receptor splice site, but also mutations within the exonic region that are also disrupting splicing. So first, I wanted to talk to you about an example that we came across early on in my project that really led us down this path of studying these splice altering variants. So here is BRCA1. It's a gene that's involved in DNA repair, and it's highly mutated in breast and ovarian cancer. And so any alterations to this tumor suppressor gene can lead to tumor development. I'm showing you here RNA sequencing reads. So here on the top is the DNA sequence. You can see by the colored bases. Below that is the amino acid sequence. And here I've highlighted where the end of exon 5 is. Here's the end of the exon junction, and exon 6 is somewhere over here. And all of the RNA sequencing reads should align only to the exon region, and they should span the intron boundary. And what you can see is that a majority of the RNA sequencing reads align to the end of the exon. But what you can also see is there's this upstream region that's utilized at a lower frequency. You have about one read here, for these 4 different samples. And so this is the normal splices that should be used. And if we look at what that donor site should look like, I've highlighted all of the bases in green that are preferred based on what that donor site should look like in the genomic sequence. And so you can see that there's 2 bases that aren't preferred, but most of the bases are preferred for this canonical site. So that's why it's utilized. But looking at this upstream site, we also see that there's a lot of preferred bases that kind of match this site. So sometimes the biological machinery identifies this upstream site and utilizes it within normal tissue. But what's cool is we found 2 samples that have a mutation just adjacent to this upstream site. And because of that, you see usage of this alternative splice site all the time. And why this is important is because this mutation was identified as a missense mutation. And normally that would be not classified as altering the whole reading frame of the protein. But because of this mutation, it completely shifts the entire rest of BRCA1 where it is no longer able to function. And so properly identifying these in a systematic way is very important for understanding tumor development. And so how do we do that? We developed this really cool tool called MySplice that's able to identify these types of events. And what we do is we identify where is the mutation located relative to the RNA sequencing reads that support the exon junction. If the mutation is located near a novel splice junction, we maintain it for further processing. If it's located near a known splice junction, we throw it out because it's the normal splice product. And then we can also do a case control comparison, where we look at where that mutation is located relative to the novel junction, but we can look in another subset of samples to see, is it a normal alternatively spliced product or not? And if it's normally spliced, we don't really care about it because it's not mutation induced. But if it's specific to the case sample, then we maintain it. And this is our subset of mutation induced novel splicing. So we scanned through about 33 different cancer types and looked at all the somatic variants. The Cancer Genome Atlas has published many different RNA sequencing and DNA sequencing samples across over 10,000 different samples. And this lets us kind of sparsely see how does this splicing alteration really affect broadly different cancer types. And when we do that, we can see many different mutations were conventionally annotated as different types, mutation types initially, but all of these are leading to splice altering variants by making a new splice site. So as we would expect, splice site mutations do make new splice sites. They disrupt the canonical site, but also a nearby site is utilized as well. But what's really important is missense and silent mutations are also leading to altering the splicing process. So these are not correctly annotated, especially silent mutations. Those would have been disregarded in our tools as not being important. So these are actually making these splice sites and completely disrupting that final mRNA and thusly the protein structure. So great, we've identified all these events. What can we do with them? Let's try and understand if there's any interesting patterns that pop out. So if it's classified as a splice site mutation, something we can look at is where is that mutation located relative to the new junction. So this would be the canonical junction, this would be the novel splice junction. So what is this distance? And similarly for a missense mutation, where is this mutation located relative to the new splice junction? And what we find is that there's a whole lot of mutations that occur within the introns of the new splice junction that's created, with a very big hotspot at this -3 position away from the new exon. So why is this a hotspot? Why do mutations normally occur 3 base pair positions away from the new splice site? Well, what we find is that a lot of these mutations are occurring at the splice site, so the mutation occurs at the G position, and if this is where the exon junction should be, the mutation at this G position can lead to the use of this nearby AG site instead. So this is the sequence context for a majority of these variants, whereby you get depletion of the canonical site, but also use of this nearby site that also looks like a splice site. And why this is important is because this third position actually strengthens binding of the splicing factor that identifies the acceptor splice site. So by having this third mutation, this third position mutated to a C, for example, it makes it a much stronger binding site for this E2AF1 protein. So that's great. We identified this really cool pattern, but we wanted to back up and look at where are all these splicing mutations located? What genes are they in? And when we look broadly, there's a lot of genes that have one associated splice site creating mutation. There's many genes that have 2 splice site creating mutations. But with this red bar, I'm showing you all the mutations in cancer-associated genes. There's lots of these events happening genome-wide, but very specifically in cancer-associated genes, with TP53 having the largest number of splice site creating mutations. So it's cool that we're picking up genes that are of interest to us that are also really important in cancer and that are recapitulated in our splicing data set. Furthermore, we see a lot of gene cancer type combinations that are very important. For example, GATA3 is highly mutated in breast cancer, and we were able to identify that in our splicing data. Same with low-grade glioma and ATRX. We can see a very similar trend. It's a hotspot in this particular cancer type. So this is good that we're identifying these common trends, but also it gives us confidence that our other events that we're calling is likely also true. So we identified all these events, but can we also look into a mini gene assay and say, if we put it into some cells, does it utilize this new splice site that we're predicting? So we did this mini gene assay where we introduced our exon of interest into a plasmid. And once we do that, we can put them into some cells and see if the cells express this alternative product with and without the mutation. So in the end, essentially what we're looking at is what is the size of the wild type product that's transcribed, what is the size of the mutant product, and what is the size of the vector by itself. So we'll be able to see a size differential based on with and without the mutation. And so I'm going to show you, I'm going to walk you through this one example we did for for BAP1. So BAP1 is a nuclear deubiquitinase that's mutated in kidney cancer. And we found 2 different samples that have a mutation. Here we have the DNA sequence, the amino acid sequence just below. Here's the known exon boundary, and here's the mutant exon boundary. And these 2 samples with the mutation at this position lead to truncating this exonic sequence by 27 base pairs. And so when we look into the mini-gene assay, we can introduce the wild type sequence, which is supposed to be 368 base pairs. And then we can see that the mutant sequence here is 27 base pairs smaller. So we can identify that this change differential does occur, and then we can sequence these bands and see if it's actually true. Furthermore, if we look at the splice score before the mutation, which is indicated up here, it's very negative. But if we look at the splice score after the mutation, it increases dramatically. And the splicing score is essentially saying how much does this new site look like a splice site before and after the mutation. So maybe that's why we see this new site utilized, because it increases the splicing score significantly. But also, we've identified it in RNA. We can also see the same thing with splicing assay, and the computational tools also support what we're identifying. We were able to do this for a whole host of genes, not just one. So it kind of looks like it's an important phenomenon that's happening, and we can also identify it in vitro and computationally. Yeah. Yeah. So those are all really cool, but what does it mean clinically isn't relevant. So a lot of these splicing sites actually make specific peptide sequences that can be manipulated clinically. So here we have the highly recurrent GATA3 mutation in breast cancer. This GATA3 is a transcription factor that's highly mutated in breast cancer. And this CA site here is deleted in 18 different samples in our data set. And because of that mutation, this AG site, which is the splice site, is completely disrupted. And because of that, you get a 7 base pair frame shift in the rest of this transcription factor. And what that translates to on the protein sequence is you get this complete end of the protein that's frame shifted. And this is a transcription factor, so it wants to bind to DNA. And without this zinc finger domain, it can't do that. But highlighting this frame shifted sequence, we can see that there's lots of different peptide sequences that are specific to this region. And because of that, we can try and manipulate the immune system to try and identify these novel mutant peptides to say, this is a tumor cell versus this is a normal cell. So this is something that we can identify. And in a few samples in this data set, we were able to identify the frame shifted product in mass spec data. So it is real, we can identify it on the RNA and the protein level. So to summarize this first part of the talk, we've identified nearly 2,000 of these somatic splice site creating variants that are present across many different cancer-associated genes and are likely having an effect towards the tumor cell development. Many of these mutations are enriched in new introns, and we were able to identify these alternative products in a mini-gene splicing assay, so they're likely true. We've also been able to identify novel new antigens that are specific to these sequences, which can be manipulated by the immune system in a clinically relevant situation. And so with that, we've identified a lot of these somatic events. So we can identify them, we see them in the genes we want, but now we want to look at the rare side. So we have lots of germline mutations that can predispose people to cancer. And we have done a lot of work in the lab looking at these, identifying rare germline pathogenic variants. And as we're classifying these mutations, we want to see, are there any that we're missing? So in our lab, mostly Kwan and a few others in the lab have led the effort in identifying these pathogenic germline variants in various cancer types. So here on the bottom, I'm showing you different cancer types. On the y-axis are different gene combinations. And so, for example, ovarian cancer, about 9% of samples have a BRCA1 mutation, a germline mutation. Similarly, you see a lot of samples that have BRCA2 mutations in ovarian cancer. And while we've identified these cancer type gene trends for various cancer types, it's important to see, are there any that we're missing that can add to this list? Because if you have a rare pathogenic germline variant, it can predispose you to cancer. So understanding if there's any we're missing is a vital question in this field. So for this analysis, we grabbed a larger subset of somatic variants to run through our pipeline. It's assumed that somatic variants are likely leading to tumor development because they're only present in the tumor cell. And then we looked at rare germline variants. So the first thing we did was we subsetted all of the germline variants in our data set. We looked at just the region that was less than 1% in the population, and that led us to a smaller subset of mutations that we ran through our data set. From that subset that we ran through, we then focused on only variants that were less than 0.05% in the population, with the assumption that if it's less than 0.05% in the population, that it's probably highly rare and may be contributing to the disease. So with that data set, you can kind of get an idea of what those numbers were here. We ran it through our pipeline, and essentially what we did differently in this second analysis is we added a annotation script. And what this annotation did was it was able to translate what all of these changes were on the RNA and on the DNA. What kinds of structures may be lost? Are we seeing changes globally in the sizes of the exons? And what was the population frequency as well for the germline variant? So with that, we identified around 2,800 of these splice site creating mutations that were in both the somatic and the germline. Looking at the distribution of those variants across different conventional annotation types, many of them, again, were in the splice acceptor, missense mutations, and synonymous mutations. So we see the very similar trends in the somatic splice site creating events, as well as the rare germline creating events. But what's important, again, is that we identify lots of missense and synonymous variants that are not properly annotated. They're actually making a new splice site, but they were identified incorrectly by our pipeline. So the first thing we wanted to look at was where are these mutations located relative to the original splice site. We know that these mutations are leading to the use of this novel splice junction, but how far away are they from the canonical splice site that should be utilized? So on the x-axis here, I'm showing you the distance in base pairs from the end, the distance from the end of the canonical splice site to the new splice junction. And you can see that I've given you a 200 base pair range here, and this shows the proportion of sites that are attributable to different types of conventional annotation. So for example, close to the donor splice site, you see lots of donor splice site mutations because they're at the donor site. But within the exon, you'll see lots of synonymous mutations that are occurring farther out into the exon, as far as 200 base pairs away to the canonical splice site. So they don't have to happen just nearby. They can also happen within the exonic region, and they could be utilized. And we see the same thing both from the rare germline front and on the somatic side as well. So we see the same trends, lots of missense out into the exon, lots of synonymous. So all these mutations are happening throughout the exon, and we really need to understand where these are in cancer. So the easiest thing to do now is look at how does that site change before and after the mutation. So what we can do is look at the canonical site with the mutation and the canonical site before the mutation and see how that splicing score changes. How does the mutation change that normal splice site, and how does it change the new splice site? So if we do this difference between the canonical post-mutation minus the canonical before mutation, if this score is positive, it would mean that it's much stronger in the presence of the mutation. Similarly, with the novel site, if we do this canonical score, or the novel score plus the mutation minus the novel score after the mutation, if this difference is positive, it means the novel site is stronger with the mutation. Now, if we do the difference between those two, the novel difference minus the canonical difference, we can get an overall change in splicing score. So this would be that the novel site is increased more so than the canonical site. And you can generally see for this group that I've plotted here that everything has increased in the positive direction, meaning that the splicing score has increased at the novel site relative to the canonical. But if we look at that for other mutation types, we can see the same type of trend. So here we can see that same thing, where for every type of mutation that we've identified, for every conventional annotation, the splicing score seems to change in the positive direction for all the novel sites relative to the canonical site. But what we wanted to understand a little bit more is why there was so little variation in these donor sites. And for the acceptor sites, what's happening there? Why do we see such large variation and a much stronger change in the overall splice score difference? So focusing just in on those two sites, here I'm showing you the difference in the novel site before and after the mutation and the difference in the canonical site before and after the mutation. And again, everything in the positive direction would mean it's stronger with the mutation. Everything in the negative direction means it's weaker with the mutation. And so for the splice donor site, what we find is that the novel splice site doesn't seem to change. Even with the mutation, there is no alteration to that new site that's used. But for the canonical site, we see a dramatic depletion in the splicing score. So these donor mutations are disrupting the canonical site and a nearby site is used. For the splice acceptor, in contrast, we see that for the novel site, there is a dramatic increase in the splicing score with the mutation. But for the canonical site, there's also a dramatic decrease after the mutation. And what that generally tells us, if we back up a little bit, is that all you have to do for the donor site is disrupt the canonical splice site, but not affect the novel site. But for the acceptor splice site, you need to both increase the new splice site and decrease the canonical splice site. So it's a two-part process. So great, we've identified all these events. We've identified this cool trend for at least the splice donor and acceptor sites. What about the final exon that's altered? Is there any relationships or patterns there? On the x-axis here, I'm showing you the canonical exon size, and on the y-axis is the novel exon size after the splicing defect. And generally what you can see is that everything is in the lower right-hand quadrant. And what that tells you is that in the presence of the mutation, the exon size tends to decrease instead of increase. And that could suggest that it's getting rid of either domains or protein structures that are necessary for this gene to function. So understanding what those domains are would be the next step in this process, and understanding why is it decreasing the exon size to lead to tumor cell development. So after this change happens, after the splice site mutation leads to altering the reading frame of this protein, we can see that we can identify if these products are in frame or off frame. If these products are off frame, where they shift the reading frame of the protein, it may lead to introducing a premature termination codon somewhere in the sequence. And there's something we know about this process in that here we have the start codon, which is the AUG. All of these lines indicate the exon junction, and we have a normal termination codon at the end here. So if a stop codon is introduced anywhere upstream of the last exon junction complex, it's expected that there's a process known as nonsense-mediated decay that should degrade this transcript. But if that new stop codon is introduced within 50 nucleotides of the last exon junction complex, or anywhere in the last exon, it should escape degradation. So it should be degraded in this case, and it should escape degradation in these cases. So we can identify where that new stop codon is introduced after translating the final altered splicing sequence. And what we generally find is that here's a measure of junction usage based on the novel site. So a higher value here means that the novel junction is expressed highly. And you can see that if I break it up into the group that should escape degradation and the group that should elicit degradation, the group that is escaping has much higher overall junction usage than the group that is expected to be degraded. So we're seeing that these mutations are altering the exon such that they're decreasing the protein sequence. If they shift the peptide sequence at all, then it's introducing a stop codon somewhere downstream that will escape degradation. So these are two mechanisms that the tumor cell is taking advantage of. So I've shown you some global trends in these splicing defects that we've identified. But what about a couple of interesting genes that are being altered because of these defects? So we've identified a few interesting silent mutations in this gene known as RAD54L. This is a DNA repair gene that is involved in repairing DNA, kind of like BRCA1. And here I'm showing you the exonic sequence just here. So this is the exon, we have the intron, and then the downstream exon. And I'm showing you the wild type sequence up here. You can see that there's 20 reads for this wild type sequence that support the novel, that support the wild type sequence, so this is the wild type exon. But I have 2 mutant samples that have a mutation at this position. And because of this mutation, it uses this new splice site. And this new splice site in both samples are used at a higher frequency than the wild type sequence seen on top here. Looking more closely at this new sequence that's used, if we look before the mutation, we can see that the splicing score is 0.98, so not too high. But after the mutation, it increases to 8.73, which is much higher. So maybe that's why it's utilized in this particular sample. Furthermore, if we look at the canonical site, that's the site that should be utilized, we can see that there's no change in the splicing score. So it's not affecting the canonical site, but it's affecting this novel site. So to try and understand what this mutation may be doing, it's involved in DNA repair. So we thought that maybe it could be leading to an increased mutation load in our patients of interest. Because if it can't effectively repair DNA, then there should be more mutations that are present within the cell population. So the first thing we looked at is the total number of mutations for each of these associated cancer types for all of the samples, and then relative to our sample of interest. So our first RAD54L sample was identified in a head and neck population. And in this population, you can see, again, for each sample, the total number of mutations. And our RAD54L mutant has a relatively higher mutation rate than the rest of the cohort, which averages around here. Same with the testicular germ cell tumor sample. It has the highest mutation rate relative to all other samples in its data set. Secondly, we also looked at age of diagnosis. So we can expect that since this is a germline mutation in these patients, maybe because of this introduced mutation that's present in all of its cells, it could have been predisposed to cancer earlier than other samples that weren't predisposed. So here for age of diagnosis, for our head and neck sample, we see that it has a slightly decreased age of diagnosis, but not too significant. Same with our testicular germ cell tumor sample. It seems to have a much lower age of diagnosis than average for this particular cancer. The last thing we could look at to try and glean if this is an important mutation for mutation load is looking at the overall mutational signature. So if we look at all mutations present within somebody's genome, we can try and categorize what those mutational contexts are based on a certain biological defect. For example, if you smoke most of your life, you will see certain genomic changes in your DNA more so than other people. And we can identify that in the DNA. So we were able to look at the mutational signature of this RAD54L mutant, and looking at, there's lots of different genomic signatures that are attributed to different types of biological effectors. But what we see is that in our RAD54L mutant, signature 2, 13, and 10 are all present. And these are all signatures that are present in samples that also have defects in DNA repair genes. So this is likely telling us that this mutation could be affecting the overall DNA repair rate in these samples. And it was originally annotated as a silent mutation. So this would have been completely missed in these 2 patients who are potentially predisposed to cancer. That was one cool case that we like, and we're going to see if it's cool. But besides that, we also did a mini-gene splicing assay to see if alternatively spliced products were formed for these rare germline events. And again, we were able to see that, and it looks good, it's legit, we like it. And so to summarize the second part of the talk, we reported a lot more of these rare germline pathogenic variants and a lot of somatic splice site creating variants as well. Many of these are expressed in cancer-associated genes, and they seem to evade degradation, so it's something that the tumor cell is taking advantage of. We learned some interesting phenomena about mutations that overlap donor and acceptor splice sites, which could be potentially relevant when we're classifying these mutations in the future. And we identified 2 interesting RAD54L pathogenic rare germline variants, which could have implications in the future for these patients if classified properly. And finally, we were able to validate a bunch of these in a mini-gene splicing assay. So I hope I've been able to convince you that in this process of going from no tumor to tumor development, we've been able to identify a whole host of mutations that weren't properly identified and were much underappreciated up until this point. And we've developed a really cool tool that's able to identify these events more systematically. And so this really emphasizes the importance of classifying all mutations in a splicing context because they could disrupt the alternative splicing process. So with that, I'd like to thank all these people in the lab. And I think I'm going to do like a two-part. Yeah. I'm going to stop and take questions, and then I'm going to do a second acknowledgement part. So thank you. Any questions? Yes.
I got a couple of questions. So first, when you stick to your slice applications to do a date in MD, but at the same time, do you think it's biased at all while you're sequencing that? Because if you're causing an MD by slice mutation, you're going to loss of RNA, we're not going to affect actually all slice mutation.
Oh yeah, that's true. So we're only picking up events that, we're biasing it. I know it's true. We're only picking up events that are either escaping it or are, I mean, I'm picking up events that are also present that should be degraded but aren't degraded, right? And I don't think that's necessarily true that they're not degraded, either that they're expressed at a much higher level and then I'm seeing some slightly lower rates. So evaluating that in this data set wouldn't be great.
And then the second question, you can see how this would work really well in your loss of function.
Mm-hmm.
Do you see anything that would look like a gain of function?
Yeah, I do see a lot in oncogenes. So the next thing we kind of want to look at is, if I look at expression, I do see a lot of samples that lead to increased expression of that particular gene because of the splice site, potentially because of the splice site creating mutation, and in oncogenes. So the next step would be, I have a couple samples where it's a germline variant in this oncogene. So I want to look at the RNA of the tumor and the RNA of the normal tissue to see if it's selected for in that cancer type of interest. But I haven't looked into that deeply yet.
So is there a correlation between having a splice mutation versus, mutational burden, total mutational burden? Are these just passenger effects where cancers that just have high mutations and just happen to have one splice?
I'm not sure about that. I haven't done a correlation between mutation burden and where you expect to see these events. I mean, they're very low frequency splice creating mutations, so I don't.
The red people was an interesting example, but I'm not sure if it's causal of the high mutation burden or if it's just a passenger.
Yeah, no, that's definitely possible, but I think it's.
I believe.
Any other questions?
It was Mike.
I know, Mike, what the heck?
I have two questions. So, possible interspecies comparison of the nucleic acid level, so at the DNA level, can you do an interspecies conservation comparison and predict missense variants and silent variants where the wobble position is highly conserved and perhaps identify mutation sites that would create de novo splice sites based on that conservation. So for example, proximal to the splice donor site, there's often a G residue.
Mm-hmm.
When that G residue is a silent variant based on your ensemble annotation, but it's also highly conserved, is it likely that that's going to be splice altered?
Okay, we can look at it.
Look, could you find potentially, so maybe your discovery is biased at the RNA level by requiring observation of alternative splicing, and maybe you could look at some of those sites in gene expression levels and identify sites that are creating aberrant splicing and also increasing non-substituted decay in those aberrant transcripts.
Yeah, that would be really cool to do. I think we didn't choose to look from the genomic side just because we did that at first and we got a lot of crap. And so,
But you'll have time before you fly to California.
no.
And so my other question was, with your splicing score, so there's a lot of tissue-specific alternative splicing, and does your splicing score account for that? And could you basically create a tissue-specific splicing score based on alternative splicing patterns?
No, we don't take that. I want that.
Identify specific tissues. Is there a grad student that can work on that?
No, I'm gonna work on it next. So that's really cool. So yeah, looking at tissue-specific effects is definitely the next way we can make this tool much better. We're only looking at, if I see any evidence of that alternatively spliced product in any of the control samples, so any other sample in that tissue type, I ignore it. So I'm sure there's a lot of events I see that are present in overall in many different cancer samples that we threw out in this data set. So developing a splicing score for a particular tissue type would be the next big thing to do in this field. Yeah.
And so I want to hear a lot more time, but are there other splicing factors besides UPA1 that you think are important? And do they have defined binding motifs adjacent to the splice donor and splice acceptor?
Yeah. There's a whole host of other splicing factors that I didn't talk about. Many of them can be mutated in cancer, but since we didn't see any dramatic mutational hotspots for those sites, we didn't choose to look at it for this data set. But I'm sure there's mutations that are affecting how different splicing factors bind to the acceptor or donor site, and associated sites that are either enhancers or silencers that are changing. Instead of more globally, they're changing at that specific site. But we just didn't choose to look at this for this set. Any other questions? Yeah.
Welcome, and I'm here to help one ask some questions.
Oh God, no.
So, in terms of this abnormal slicing, did you find any difference between the benign tumor from the malignant tumor?
But not, whoa. Yeah, maybe. I mean, that's not a benign tumor, so just something that doesn't lead to, yeah, that's not something we looked at here, but you might, I'm sure alternative splicing is all kinds of crazy different tissue types, so I'm sure you could see very specific changes in a benign tumor that aren't present in a malignant tumor and vice versa. But I didn't look at that too. No. Okay.
So first, I would like to thank the sitting. So first, I want to thank Lee.
Oh, come on, Clay.
Yeah. So Lee has been a completely supportive mentor this entire time. See, she's supporting Chris when he hurt his wife. Were you pushing him? I don't know. He probably hurt his leg or something. But it's been so much fun to work for a woman who, it's been cool working for guys. They're all cool, but it's been really inspiring to see how she interacts with people in the community and to try and grab onto some of those characteristics and try and kind of emulate the strengths she has in being a mentor. But also she's been a lot of fun, very nosy and wants to know everything about your life, and it's been fun to try to. She loves it. It's fine. We like telling you too. And it's also fun because she let me have a lot of freedom in the lab. If I wanted to change something or do whatever, she would usually let me. And that was pretty cool. When I first joined the lab, it was very there was a lot of staff scientists and postdocs, and they wouldn't really talk about the biology behind it. And I was like, what the hell are you people talking about? And she let me ask questions, very dumb questions, and try and figure out what the heck was happening. And I think we have a much stronger population of women in the lab now, which is legit. And I love that. And this lab is ridiculous. Look how cool they are up in the upper corner. We have a really good family in this lab. I think it's why we keep attracting all these awesome students. They all dress up for Halloween, which I love.
All those onesies are definitely not Halloween, but we just have them in lab. I only have a handful of photos because I finished this crap last minute, but like it's been so much fun being in this lab with this group of people, and I couldn't have asked for a better group to suffer with for the last 5 years. It was fine. And then of course, Quarine, we have been like a really fun bunch. We all joined together. Of course, Bailey's here secretly too, but also we hang out way too much, and it was cool having 2 really awesome other friends in the lab. We went on trips together and hung out like at the end of the day, most days a lot, and it was a good time. I also had a crap ton of other friends. These are just all the pictures I could find. Apparently I don't take pictures of people, so I couldn't go through all your Facebooks and find out my stories. But like there's some people who went on marches with me, which was a lot of fun. I think the first class from DG students I have to be thankful for because you all studied with me my first year. Like when I first got here to watch you, I was like, crap, I'm not going to pass because none of you guys will study together. I can't study by myself, but I somehow tricked you all into doing that. And I passed and stuff. And then thanks again to like the WashU community for like doing presentations in ridiculous outfits. Like I've appreciated that so much, and I think some of the students who come here also appreciate that. And we have a super great group of softball, yo. Like we have a team. Go join it.
Go reference.
What? We got you. Whoa. And yeah, we've had a very good time throughout the year. Also, my floor is legit. We have a really cool oncology floor that I'll dress up for Halloween. And all the staff on our floor are amazing. They'll just be like laughing half the time, and I'll have to run over and see what they're talking about. They've made everything so much nicer because I know it's it's a very happy floor in general. There's very minimal cussing.
They're here to support you.
Yeah. And of course, there's other like DBBS people that I totally could not have in this presentation, but like Sherilyn, Laura, Patrice, there's Melanie. There's so many people in the DBBS community that have helped support me throughout this process, and I really couldn't have done that without having that group of people that were there to like bring me up when I was feeling really crappy. And like going and feeling like a family was so important in this process. And of course, Jim. I like totally would not have made it through if it wasn't for you. I've never like met someone who's cared so much about like the students that aren't his own blood, and I just don't I don't understand how you do it and how you're still doing it, but like I've appreciated it so much, and I'm sure the rest of the graduate community does too. I also wanted to thank some family. So this is my mom's side and some other randos that couldn't make it, but have been very supportive the whole time. We'll talk about those people a little bit later. But like my mom's sister was able to make it, which is really cool. And then, all my bitches. So we have had a lot of fun throughout the year. Like Laura, thanks for being my roommate for a bunch of years, and like that's good. It's been really fun to like go home with someone who doesn't want to talk about science necessarily. Like we don't talk about science, but like very politically active, so that we can like be happy and sad at the same time. Like we'll end our Sundays with John Oliver, and we're usually really pissed off, and I don't know why we do that, but Shamini, it's been so much fun having like a Biffle in the lab. Like we'll hang out all day, gossip all day, do work, too. And then at the end of the day, we'll go drinking, and we like don't get tired of each other for some reason, and that was good because I was worried. But maybe like a little separation will help people think we're not sisters anymore. Really cool. Jeanette, thanks for being like, I know you're not normal, but like the most normal. Like it really helps like to round the group out a lot. And thanks for coming today. That's so cool. But I'm excited for when we're gonna be in San Francisco together. I'm like, I'm ready. You know, we're gonna like start doing stuff. But we've been on so many adventures together. And then Bahana we found somewhere along the way. And she's just completely integrated perfectly into our group. And like I'm just really pissed that we all like I like want us to all be in the same city forever because it's like my crew. But also I think that we're gonna be good friends for a long time because we know way too much shit about each other. Jason. Oh, it's gonna move. It's gonna move. We've been together for like way too long, in a wonderful way. But thanks for like letting me go away to grad school for like the experience. And like I think you're the reason why I'm in genomics. I remember like going over to your house, I would find these C++ coding books, and I would try the Hello World crap. And I remember writing my first code and thinking, whoa, this is so awesome. And I'm pretty sure that's why I ended up in this field. So thank you for that. I like appreciated you supporting me all throughout undergrad, and then going away to grad school. It's been very important for me. And then my family, and a lot of them are here. So we have my dad holding the puppy up there, and my brother holding the puppy up there, and my grandma over here, who are all here over here. And then we have my, this is my crew of this Eric and Sonali, who are also here, and Shiromi. And we've been like a unit forever, I think. We all grew up together. We're all homies. Stop crying. I haven't said anything yet. And I think we've been through a lot. We've gone through a lot of like deaths in the family and a lot of like new life as well. And we have completely grown as a crew together, and it's been completely amazing. I know that whenever I come home, they're my home base, and it's just been like it's been cool to see how like our interactions have changed as we've gotten older. But also, I know that we're going to be a very tight-knit community or group for a while, and I greatly appreciate that. And I think it's very serendipitous that I'm doing my defense on my mom's birthday weekend, because the reason I'm in this field is because when I was younger, my mom was diagnosed with cancer, and she, like watching her go through that process was really difficult as a young child. But I also noticed that even when people lie about their history of cancer, for example, or when families don't talk, you can't share whether there's a family history of cancer. But the genetic DNA does not lie. You can identify variants that predispose you to cancer. You can identify changes in the genome that can lead someone to getting cancer. And that's why I'm in this field, because I think it's so important to be able to identify these events early on. And if we can see if we can help someone else diagnose that early on. And so with that, I'd like to thank everybody here and my committee for being so supportive in this process. And thank you all for listening.
Thank you all, and I think we will, what time?
4:30.
4:30.
4 o'clock to 4:30.
So we have a celebration and on the third floor, activity building at 4:30. You guys are welcome to join us.
Let's go.